Today's topic is DNA replication. So the process is duplicate the DNA. Before the cell division need to happen, DNA need to be duplicated. So before the cell, we call the mitosis. In our last unit, we'll talk about mitosis. And the process to make double DNA so they can duplicate, they can duplicate first. That's the DNA duplication. So when the cell have to divide, because, well, you need to heal, the cell need to divide. Also, you can replace damaged cells. And also when you grow, you need more cells. So all these reasons, you need to have cell division. And sometimes your reproductive system produce gamete. We will talk about the meiosis in next unit. Produce egg, produce sperm. So all this, you need to have cell division. And for some simple creatures, they only have one cell, the single cells organisms. Their cell duplication means they reproduce. So they reproduce asexually. So all these are the reasons you need to have DNA duplication. Okay, so let's start the DNA duplication. So DNA duplication, you need to split the DNA into two and use each one of them as a template to produce another one. So that's why we call it semi-conservative, because every time you duplicate DNA, now you have two strands DNA, and each one of them is one old, one new. So we call it semi-conservative. That's how you replicate DNA. You always have one old and one new. And these double helix, they are identical. So if you take this strand, read from top down, and this strand, read from bottom up, eventually you'll find they're all identical. So you have the double informations. Okay, so now let's look at the DNA. They stay inside the nucleus. Most of the time, they are loose structure. I call them spaghettis. They look like spaghettis, but actually their name is called chromatin. So those loose DNA structure is called chromatin. And you're more familiar with this term called chromosome. Chromosome is they are condensed, especially when the mitosis, the cell division need to happen. You need to condense them and line up in the center of the cell, and you can pull them to two ends. So those condensed one is called chromosome. And to condense them, you just, DNA is a long sequence. You take those DNA helicase, histone protein. There's a protein, you just wrap it, use those DNA wrap it. So now you can condense them together, become the chromosome. And the chromosome have some names of this structure. The end is called the telomere. So each chromosomes in the end is called the telomere. There are some studies about telomere. They say it's associated with your life age, because every time when the cell divide, the telomeres get shorter and shorter. And when they get too short, they just could not divide anymore. So the cell will die. And they just look at your telomeres length, and they can predict your biological age. That's the telomere. And the central part is called the centromere. This is centromere. And each one of them is called the arm. Okay. So this condensed, this is the chromosome, and loose is the chromatin. So usually those DNA are in a loose state. We call them chromatin. Okay, so let's review the DNA. DNA is made of double strand. Each strand is made of a nucleic acid sequence, right? Nucleotide. And they just twist, and then they use the hydrogen bond, which is a weak bond, to glue them together. So those two strands can glue together. So they are very stable. And when they glue together, the structure need to be maintained because there are totally 5 different nucleotides, A, T, G, and C. And DNA use 4, A, T, G, C, U, totally 5. But DNA use 4. DNA use A, T, G, C, these 4. And 2 of them are big, 2 of them are small. So A and G are big, and T and C are small. So when they pair, they have to use a big one pair with small one. So the DNA double helix structure will be the same size from the beginning to the end. So it turned out the A pair with T and the G pair with C. So that's how they pair together. And that's nucleotide. So they have a nitrogenous base and they have a sugar. So if you remember what you learned, this is the 2-deoxy ribose. Yeah, because does not have the OH, only has H. And there's a phosphate group. So that's the nucleotide. Okay, so let's start with the replication. So you want to duplicate the DNA. The first thing is you need to split them. And once you split them, you have to use the pin to pin them so they won't glue back together. Those hydrogen bond won't form. So you need enzyme. This enzyme to unwind, to pin them in the place is called DNA helicase. You need to know the enzyme because I will ask you the enzyme's name. So DNA helicase is unwind the double helix during the DNA replication. Okay, what's the second one? So now you unwind them. Say, well, this is a very short DNA sequence. We use this as an example. So originally A pair with T, G pair with C. Okay, now you just split them open. Those hydrogen bond, and you split them open. Now you have 2 single strand by using a DNA helicase. Okay, now what are you going to do? You're going to use each one of them as a template to produce another one. We say semi-conservative because each one are going to become a template. Okay, and then how do you produce a new one? You know A pair with T, C gonna pair with G, T gonna pair with A pair with T, because, well, you learned a big one need to pair with a small one. So that's how they produce a new strand. So those dark blue, these are the old strand. And then you produce those light blue, these are new strand. So these are new strand. And the enzyme to put those new nucleic acid there is called DNA polymerase. That's the enzyme. So DNA polymerase and create a new strand. It's called the complementary DNA nucleotide. So A pair with T, C pair with G. And on this strand, okay, it goes with T, G, A, T. So you know, okay, you're gonna pair with A, and G pair with C, A pair with T, T pair with A. Just keep going down. So now you have 2 new strands, and those 2 new strands is actually one old, one new, those double helix. And that's the DNA replication. So after you duplicate, DNA is a long sequence. So before they release them, the third process is proofreading. So you need to proof, especially you are making a new sequence, you're going to make a mistake. So you have the proofreading process to avoid mutation, because if the DNA is wrong, the RNA will be wrong. And when the RNA is wrong, the amino acid, and which is a protein they're gonna produce, will be wrong. So the third process before they release those 2 new strands is proofreading. And after proofreading, you have two new DNA sequence, and that's the DNA duplication.